So, a very good afternoon to all of you, and we welcome ma'am, Professor Jyotsna. Today we are at the last day of our lecture series, which was a 5-day event, and today we are having two sessions. And first session is from Professor Jyotsna Agarwal. So, very short words and introduction about ma'am. That she is additional professor, consultant, editor, secretary, positive psychology and integral machine medicines. She's for integral medicines. She's faculty in charge and for VIPRA, Vedic Indian Psychology Research and Application. She is from Department of Clinical Psychology, NIMHANS. And she brings together a unique blend of expertise in clinical psychology that pertaining to NIMHANS, MPhil from NIMHANS, and positive psychology, where she has done PhD on happiness at NIMHANS and postdoc on positive parenting at Washington University School of Medicine, USA. She also has a background in yoga and Indian psychology, postdoc at SVYAS, which is Yoga University, and various other certificate courses at IPL and Sakshi, etc. In addition, she has worked with a large community in rural area while doing PGD in rural development and work in community at Tata Motors Jamshedpur and Community Empowerment Lab Lucknow, and with youth at IIT Patna and corporate at Tata Motors Limited Jamshedpur. She is currently working on the application of Indian psychology for therapeutic and promoting goals that has several research and publication in these areas. So these are the few words which is just briefing about the ma'am. But as we know that ma'am always been a very great mentor for all of us, and she has been contributing her knowledge for bringing our projects into the reality. So with these words, I welcome you, ma'am, and thank you for your time and your session today. So stage is yours, ma'am. Please. One more thing, ma'am, we have just got some international visitors, delegates over here in CDAC Delhi. So in between, I have to just go for a minute. So in between, I will be leaving for a while. In between, Soumya and Akanksha would be there to host you. So I will take your leave for 10 minutes in between, ma'am.
Sure. Thank you. Thank you so much, Dr. Priyanka, and thank you for inviting for this talk. And although I've not done too much of work with the technology, I have always been interested in this and how we can use it for mental health. So I'll be presenting what I've understood and what I see the potential. And it would be an interesting area for collaboration in future. So with this, I'll share my slides. I hope it is visible.
Yes, ma'am, it is visible.
And in a slide share mode?
It would be coming. It is not coming as of now. It is in a just running mode. It's not in execution. It is in execution mode now.
Okay, great. So I'll be focusing a little more on the emotions part. So emotions are our reactions to events, situations, and all. And when we talk about emotions, basically it is a mix of components. So there is a part which is about subjective feelings, like one's own unique experiences of things, whether it is feeling pleasant, unpleasant. So maybe if you think how you're feeling now. So some of you might say that, well, I'm feeling comfortable, or it is post-lunch. So I'm feeling relaxed, or I'm feeling a little tired, or something like that. So these are the subjective feelings. There's also physiological changes associated, which is like the heartbeat, or when people feel anxious, sweaty palms, etc. There is also cognitive processing, whether what are you thinking about, how you're thinking, whether you're thinking in a narrow manner, for example, which happens in negative emotions, or in a lot more broad, often in the positive emotions, as well as there is a part of the emotions which is about the behavioral expression, which is about whether you want to approach something, go do something, go out in the world, or else avoid something. So avoid a response towards something, want to run away, go away, etc. And different theories emphasize different components of these emotions, and some also emphasize the role of culture and how it is important in one's expression. So most basic emotions have been found to have some kind of sensations, elevated sensations, especially in the upper part of the body, like the upper chest area. And along with that, changes in the breathing, heart rate and all. And the head area, which is physiological changes in the facial area. And a feeling of sensation in the body as well as in the mind due to some kind of emotional event. So, for example, if we talk about anger, there is a negative emotion with like a lot of intense feelings, which is often seen in people feel that in the head, chest, hand, etc. Similarly, if people talk about their sense of fear or anxiety and all, these have different set of sensations. So common symptoms can be fast heart rate, hot flashes, etc. On the other hand, when you go to the depression, there is low activation actually. So if we see that from the as compared to the neutral emotion, even there is further lower activation in the extremes of the body, like in depression. So for example, hands, legs, etc. As you see in these maps, which is like one very interesting study done on the emotional map in the body. So there are a lot of models to understand emotions, like it can be seen as independent categories, or you can say discrete emotions, and there are various common or popular theories around it. I wonder if you have heard about Paul Ekman's model of 6 basic emotions, which are happiness, sadness, anger, disgust, surprise, fear. Another common and very popular model is Robert Plutchke's model, which is 8 emotions in opposite pairs, which because they are opposite, you can even make a wheel around it. Then there is Orthney, Clore, and Collins' model of 22 discrete emotions. The other way of be that it is a continuum in relation to each other, and there are various kinds of models around it. Some of them are most popular one is which who gave a circular 2-dimensional model of on one side you have a feeling of pleasant versus unpleasant, and other side you have feelings of arousal or activation and deactivation. So for example, if you see anger, it is an unpleasant emotion. Along with that, there is an activity component to it, there's an energy component to it. On the other hand, when we talk about sadness, it is again an unpleasant emotion, but there is a low arousal component to it. So there, in that sense, all these emotions can be put into this continuum in some place or other based on these 2 dimensions of pleasantness, unpleasantness, and activation, deactivation. Further, another dimension of dominance, or how much a person has control over these emotions, was added later by Russell Mapp, right? Wheel of emotion also have been added later, like balance and arousal, which can also make it, as I said, that concentric circle. So as we had seen in the last picture, so there is like a concentric pictures here. So why are emotions important? Studying emotions are important because it is a very closely part of our life. It has adaptive value. It has helped us in survival actually. So if you see, negative and pleasant emotions are very, very closely associated with our immediate benefit in terms of survival. When we have an experience of negative emotion, we want to avoid, run, escape, etc., or fight back. So when these emotions come, a lot of people have various kinds of responses. So one of the initial studies, which was continued being very, very dominant in the field, was fight flight response. That during a stress, when we have a stress feeling, we have a certain stress responses being released in our body, which gives us short-term strength and energy, which helps us to run away from a situation or fight a situation. However, when it remains for a long term, and that is why long-term stress leads to poor health and well-being. In recent times, there have been some newer models. Very interestingly, this fight-flight response continued being dominant for almost 80 years, 85 years. And then in 2000, Taylor and her postdoc looked into the data and said that, well, not everyone responds like this fight-flight. Sometimes we look into connecting with other people, talking about it, sharing our feelings, etc. And then they said that let's look into the original study, and they found that the most of the original fight-flight response study was actually based on animal model, which were rats and male rats. So they found that they did other studies, and they found that under stress, females of the species often focus on taking care of young, finding social support, which is called cooperative breeding. So that is related to also release of oxytocin hormone. And this model was known as tend and befriend way of coping with the stress. Another model about emotions, coping with stressful emotions, are freeze, flop, fawn response. Especially this happens when there is a trauma. One may feel like a cold, numb, play dead, which is called a freeze response. or become very agreeable to another person, especially those who are in the dominant position, or become very helpful. So that is called the fawn response. If the fight flight is unsuccessful, or people perceive that they cannot fight back. This was about a little more about the negative emotions. However, we also have to look into a little closely positive emotions, because it's not always that negative emotion can become a template for studying positive emotions. So positive emotions are in fact often linked to one's wish to continue or even start engagement with an activity, as different from the negative emotion, which is related to wanting to avoid something or stop something, run away from something. Here we want to start or even continue or go and grasp on something. Positive emotions have been difficult to study, and not before 2 decades there was not many studies, and therefore the field was dominated by negative emotion studies, because these are less clearly differentiated, less researched. And in recent times there has been, last 2 decades, there have been a lot of studies about it, and full richness of life remains in them. Some new interesting models have come up with related to positive emotions. It would be good to understand them. One is about how it is beneficial for individuals as well as collective well-being. If we look into when we are looking at the map of emotions in the body, we see that happiness, for example, a positive emotion, leads to activation in the whole body, which indicates a physical readiness or heightened communication between body and brain. Love, another positive emotions, affects the body with activation, except the legs part, and it leads to also heightened sensation in the reproductive organs more strongly than happiness. One of the important finding which came up was that positive emotions can actually correct or undo the effect of the after effects of a negative emotion. And that leads to a spiraling out of phenomena, which is basically that when a negative emotion happens, there is certain kind of effect on the body, which are detrimental to health. On the other hand, when there is a positive emotion, it can actually undo that detrimental effect of negative emotions, and that's why it's called the spiraling out phenomena. So, for example, experiencing positive emotions soon after a high activation negative emotion, for example, anger, led to faster cardiovascular recovery. So we also have to look into how positive emotions can make us resilient. So it can transform people, lead to upward spiral. But how? So, for example, there is an experience of positive emotion, it can lead to thinking in a broad way. And our actions become a lot more varied. And in fact, you can say it can become more, have a larger repertoire or range. So when this happens, it can lead to building of resources. So for example, if imagine a happy child goes around, plays around, talks to people and all, and there is a building of resources. When they go play, there is a physical resources being built. When they explore the environment, in fact, intellectual ideas build up. They connect with other people, social relationships build up, and all of this leads to the transformation of people. And that can in fact lead to more positive emotion experiences. So this becomes like a positive cycle. So for example, positive emotion like joy, interest, contentment and all can lead to widening of those momentary thought action repertoire, and that can lead to more of problem solving, new information gaining, coordination, strength at the physical level, resilience, optimism, etc. at the psychological level, and social bonds, making new friends and all at the social level. And this makes a person become more creative, more knowledgeable, resilient, etc. On the other hand, if we see humans do have a negativity bias. So because negative emotions signal a problem or a threat, which immediately captures our attention, and also because cultural factors, social factors like that can give a lot of attention to whatever happening is negative around us. So positivity is taken for granted, not considered many times. Right to acknowledge, appreciate the positive. We kind of take it for granted. We ignore it. We don't want to maybe build someone's pride, and that's why we don't appreciate it. And sometimes it is seen from an anti-intellectual lens that positivity looks like a superficial thing. On the other hand, a variety of brain regions have been studied depending on specific emotions in this area. And we feel that it's very important that our brain is to be trained to have more positive or calm emotions as compared to the negative emotions, and the intensity of it also has to reduce the negative emotional intensity. So for this purpose, it's important to assess the emotions, and especially we will look into how do we do it in the digital era using the digital technologies. So understanding emotions are useful both for the person who is experiencing it as well as other people. It can be used in counseling, education, even in the robotics, to make a smart collaborative service robot or even social robots. And it is also increasingly used in marketing or entertainment industry. And other potential areas can be medicine softwares, diagnostic softwares, etc. So when we assess emotions, one of the very common thing is subjective feelings. Given that subjective aspect of the emotions is one of the most important element, which is commonly asked by asking questions to people. It can be done by using various kinds of scales, for example, positive, negative affect scale, emotion regulation scale, etc. It can also be done like online. Cognitive aspects of emotions are often assessed in the clinical setting using various kinds of scales. For example, one can be dysfunctional attitude scale, or it can also be used using the text analysis. Behavioral expressions of emotions are more observable, easier to understand. However, in the clinics, it can require behavioral analysis guided by various theories to understand what is the reason behind, what is the cause behind a certain kind of behavioral expression. Physiological changes are another aspect, and it can be either can be very noticeable to someone who is experiencing, or some of it which are related to release of certain hormones in the body. Person who is experiencing them also may not be able to notice them. And these are most amenable to use of automated emotion recognition using variety of technological tools. So we will look into some of these tools. So, for example, EEG is one of the non-invasive technique for recording electrical activity. However, it is still little uncomfortable, where the electrical activity of human brain is recorded using electrodes. And the idea is that response to various stimuli are measured, analyzed by 5 frequency ranges, delta, theta, alpha, beta, and gamma, and it indicates different emotion states. However, it needs special devices, expertise, expensive, and it's uncomfortable to use in daily life. So these are a lot of limitations of the usage of EEG for emotion assessment. Respiratory rate analysis is another method where velocity or depth of respiration can be measured because it varies with the human emotions. For example, normal calm state may have 20 times per minute to versus aroused state 40-50 times per minute. And it can range from excitement related to happiness, anger, afraid, fearful emotions, tension, relaxation, or a calm emotion. It can actually be assessed using simple timers, specified softwares, etc., to measure exhaled air, fluctuations in air humidity, temperature, air pressure, carbon dioxide concentration, oxygen concentration, body movement, etc. But this is less popular due to data not being very precise and can be affected by various other factors. While non-contact measurement is possible because a body can be observed using video, thermal cameras, etc., but method needs some methods of respiratory analysis need contact sensors and can be uncomfortable too. Other ways of assessing its emotions using technologies like ECG, for example, which is another non-invasive method to tap electrical activity of the heart in real time using sensors on the body, can be used for emotion recognition because association of heart activity with different emotions has been noticed through different sensitivity to specific emotions. But this is a complex process. It doesn't lead to a contactless assessment and or an instantaneous emotion recognition. Another method is more popular nowadays is heart rate variability by measuring slow versus fast heart rate, which is the gap between the parasympathetic and sympathetic nerves, which are influenced by emotion, stress, exercise, along with many other factors. And one may assess emotional states. So, for example, low HRV indicates relaxed state, high HRV indicates possible stress or frustration, can be measured by ECG, but also by something like a photodetector or PPG, which is often used in wearable gadgets, where touching the active surface of the sensor for a few seconds is enough. For example, touch screen of a common smartphone and all. So these are the common things. Another one is also galvanic skin response, or GSR, or even electrodermal activity, which is known as skin conductance, which is a continuous measurement of electrical parameters of human skin, usually skin conduction using 1 to 2 sensors on the skin. Changes in emotion, arousal level, sympathetic nervous system can lead to sweat reaction, which can actually change the salt in the human skin, in the surface of hands or fingers and all. And this in itself can change the electrical resistance of the skin. So that is the theory behind it. And GSR signal amplitude is associated with stress, excitement, engagement, frustration, anger. Other attention grabbing stimuli, attention demanding tasks also can lead to increase in the frequency and magnitude of GSR. This data is easy to get, simple, low cost, easy to assemble, wearable, and it can analyze it with fewer raw data, but it doesn't provide information on balance level, whether it is a positive high arousal or negative. So skin temperature measurement is another method which is related to human heart activity and sweat reaction, which is then influenced by autonomic nervous system. There is good correlation between emotional state, skin surface temperature, fingertip blood flow. And it has been seen that stress, anxiety, anger, embarrassment, various kinds of emotions, hostility, guilt, etc., can lead to decreased finger temperature. This is usually assessed either by contact or non-contact methods. Semiconductor sensors for the contact, and face or full body thermal imaging for non-contact. Non-contact can be easy, popular method, though it is less accurate and lack of information on specific emotions is one of the barriers. And they're not so often used in the studies. EMG is another technique for evaluating, recording the electrical potential generated by muscle cells. Can be used to find the correlation between cognitive, emotion, physiological reaction, etc. And can be surface or skin electrodes, or it can be inserted electrodes also. So this needs a simple analysis of a signal, can identify or separate emotions, good technique to detect strong emotions, but not very often used because it is not very comfortable. Needs skin needs to be prepared clearly, the skin needs to be cleared, shaved hair, etc. And a lot of large area of body is required, like a bigger measurement area. There is an insufficient signal noise ratio also. So in this hypothesis that response to various stimuli, non-contact recording of videos can also be used to get the same idea about the efficiencies. EOG is another technique for measuring the eye movement, which detects eye blinking associated with stress, surprise, fatigue, concentration, drowsiness, etc., and can be used both by contact or non-contact measurement. Non-contact is usually the video camera or infrared cameras. And it is less precise because other factors may interfere, but it is easy to use. And it can be used by using wearable eye trackers, headsets, screen, and specific softwares. And looking at AI in the field of mental health. So AI technology has also been used in and especially in last maybe 5 to 10 years. There's a huge amount of research in this area. How AI can help in diagnosis as well as treatment. So we'll look into some of these aspects. For example, AI can help in early detection of mental health disorder by analyzing the large health records or speech text analysis, or it can lead to predictive models or improved treatment outcomes through those models. It can use in treatment, can help in personalized treatment plan, which can work very well for an individual who is not who is not responding to the usual treatment. And it can also be used for virtual therapists and chatbots, which are improving on it. In fact, you can say every day there are better versions coming up. So some of the things which are related to this is, for example, sentiment analysis, which is identification of feelings and attitude towards something, and emotion detection, which is the emotions of human language, which is useful in detection, for example, in The limitation, for example, related to language, where there is a short text, etc. And emotion dictionaries are also not very, you can say,
Ma'am, I'm getting some
hello.
problem in your voice. It is not consistent and it's breaking a lot. I think, is it this with me only or it is with everyone?
No, my voice is
I got
breaking.
your voice is breaking. Should I stop the video and see?
Yes, ma'am. Yes, ma'am. That would
I've
be.
stopped the video.
Yes, ma'am.
Maybe this is.
Okay. Okay. Yeah.
So inadequate emotion dictionaries, etc., might be present. So use case, as I was saying, that BoBot is a chatbot, a mental health app, which analyzes user text inputs. If they are consistent, it actually provide guidance.
Hello,
or
ma'am.
recommend professional help. So this is one of. Hello. Can you hear me?
Yes, ma'am, it's still breaking the voice.
Oh.
It is quite breaking, ma'am. Means we are unable to comprehend properly because lots of breaking.
I'm using the NIMHANS this internet, because mobile would further be worse actually. Should I go slow or should I join again? What would you suggest?
Would it be possible if you can join again, ma'am? That would be better. Suppose.
Sure, I'll do that. Oh, hello, can you hear me now?
Yeah, ma'am. It is much quite clear and louder, and this is good now.
Sure. Okay, I'll share the slide and I'll switch off the video again.
Sure.
So I was giving the example of a AI-driven mental health app called WoBot, which analyzes user text inputs and sees if there is a consistent expression of sadness, hopelessness, despair in the language of the chat. It actually provides guidance or recommends certain kind of professional help. So this is an example of emotion detection. Facial expression analysis, another option, which is like now often with computer vision, data on micro expressions, subtle changes in facial features can lead to insight into an individual's emotional states. So with AI, the precision is improving, can be used for remote mental health monitoring through video consultation, mobile app, and it can give an early warning sign. leading to a timely intervention and support for the patient. So one of the company called Affectiva, which works in the area of emotion AI, has developed facial expression analysis tools, which is being researched currently. For example, facial expression of individuals with depression for early diagnostic markers. Analysis of voice, gesture, body posture, etc. is another area, which is like now where emotions expressed through facial expression, voice, body posture, gestures can be tapped by recent advances, and that can lead to insight into a person's mental health. So, for example, voice analysis can detect alterations in speech pattern, variation in pitch, tone, rhythm, etc., which can lead to insight into someone having anxiety or depression and all. These are non-contact measurement, fairly reliable result is present, can be used in combination with other techniques for both individual as well as group-based emotion recognition. However, there can be limitations, especially, for example, for body posture, gestures and all. There can be many different reference points. Only strong emotions, which last some amount of time, can lead to noticeable changes. And there can be effect of environment, presence of large data, reference pointers, etc. And some of this can be covered by the clothes also. One use case is Cogito, an AI platform used in telehealth services. So given that there are patients calling or people reaching out to them, it monitors speech changes during the therapy. And any change in pitch, tone, rhythm, which might suggest anxiety or depression, so they alert the therapist to address them in the session. Predictive modeling, which you might be, some of you at least, those who are coming from technology field would be aware that we consider innumerable data points such as genetics, environment factors, lifestyle choices, social determinants, etc., and wearable technology data, mobile health data, health record, etc. One very interesting finding long back when there was a lot of enthusiasm about genetic studies was there, almost a decade back. A lot of studies were done with a lot of optimism, and then they finally found that there's no single genetic cause for any mental illness, because they found that it was multi-causal, multifactorial. And there was one of the good findings which was there, was that certain particular genes, when it interacts with certain kind of environmental factors, for example, childhood trauma, can lead to certain kind of mental illness or possibility of a mental illness. And therefore, it's not just that a particular gene is there, but also a particular environment. For when a person comes in, you have so many data points. So in this kind of situation, AI can help to understand individuals at risk for mental illness, their treatment response. Certain people may not respond very well to XYZ of medication, and they may need a certain kind of treatment or a combination of treatment. AI can also tell, based on these data points, about disease progression, or it can help in integration with a person's overall health profile. So mental health issues with the overall profile, which can improve a variety of decision making at every stage and can lead to a more holistic approach to healthcare, and personalized healthcare also, which is becoming more and more important. So a couple of use cases, for example, Google depression screening tool. So if a user searches for depression related terms on Google, it can actually prompt them to screen or test themselves, assess themselves using PHQ-9, which is a questionnaire for depression and very widely used in public health. And then they can give guidance on mental health. IBM's AI-based Watson for drug discovery is another use case where it analyzes genetic and chemical information data set, and that can lead to identification of potential drug candidates. This is especially important because it was seen that for decades, there was not much of drug-related improvement in the field of mental health, because it may not be very profitable. So those companies would investors in money or same amount of for a very long.
I'm so sorry to interrupt. Again, the voice is starting.
Which was happening.
Ma'am, we start again.
Okay.
At losing your voice. I think something had happened again. So it is again breaking a lot. I don't know what would be the reason.
I'm not too sure. There is a lot of rain in the background. I don't know whether that is leading to some kind of sound issue.
Yeah, that may be the problem. This is not only the noise in background. This is about that not getting clear words itself. So that is somehow means it is giving us a cognitive load to understand and try to comprehend. Means we are not able to understand the sentence itself in between.
Okay. Huh. Should I rejoin?
No, ma'am. I think you should continue. When you are talking, it is coming very clear now.
Okay. So coming back to mental health platform, there is something called Ginger, which utilizes predictive analytics. For example, someone having high stress, poor sleep, social withdrawal, and identifies and reaches out to those individuals who might be at risk of developing certain mental health conditions and provides a mental health coach to them. So as we see that there are variety of ways to do assessment. No single method is ideal for all situations, and multimodal analysis might be a better option. Now coming to management of emotions. So what happens when we repeatedly get stuck in negative emotions? We may feel overwhelmed by negativity, which can become very automatic. Like, for example, a person can think, I'm no good, other people do not appreciate me. They can think too much, ruminate, or overanalyze without finding or reaching any solution. They may also feel that they are not finding or not even trying out techniques which may help, or give up too soon because they feel nothing works. Sometimes such people also pick up quick over-the-counter solution, the easy and tempting ways of managing an issue, which can be also unhealthy. For example, drugs, alcohol, and they may not give chance to long-term solutions. For example, something like underlying issue can regularly trigger emotions. And one of the another important thing is that people sometimes do not give importance to developing their capacity to stay with unpleasantness, which is like distress tolerance, and very important in a life where we continue developing and progressing. So emotion regulation is about freedom to feel and let go of emotions without getting stuck into them. So some of the common regulating emotion regulation strategies, for example, for managing negative emotions are like changing our thoughts, thinking something else, thinking about same situation in different way, thinking about cause, implication, long-term solution, imagining something different, pleasant, etc. to reduce distress, and changing our behavior. For example, focusing attention on another activity to distract themselves, attention to another activity to cultivate another feeling, working on solutions, sharing with others, and by being mindful. So, interventions for emotion regulation can actually, when it is very intense negative emotion, it can be required to undergo counseling or psychotherapy. Because these maladaptive emotions can lead to maladaptive ways of handling them and can lead to various kind of mental health issues or even physical health issues. So one may look for professional mental health support for these, and different schools of psychotherapy are present, which may target more or less different component of emotions, although most amount of the work is integrative in its approach. On the other hand, positive psychology interventions especially focus on cultivating the positive emotions, positive thought related to that, positive behavior, or and need satisfaction, and it can lead to more better well-being and better outcomes. A lot of interventions are also present for this, which can lead to cognitive strategies like thinking positive emotions in one's mind and self-monitoring of well-being. Practice of optimistic thinking, it can relate to practicing emotional skills like mindfulness, acceptance, savoring. It can also be related to various kinds of positive behavior, basically like enjoying hobbies, engaging in enjoyable activities, or expressing gratitude, engage in acts of kindness, or using strength. Use of digital technology for emotion regulation is also important. CCBT or computerized version of CBT. With children and
Ma'am, request you to please re-log in again.
change in attitude. Okay, I'll do that.
Sure, ma'am. Sure, ma'am.
Hi, Dr. Priyanka.
Hello, good afternoon, sir. Actually, there was some voice breaking from Dr. Jyotsna. So we asked her to please rejoin so that we can be able to hear it, because it was there was loss of voice inability for us to understand.
Actually, the topic is a very interesting topic, both the one you have
Yes.
sent. So I was very, very strongly motivated. Otherwise, I am in the Delhi roads on my way to the airport.
Okay, sir.
Yeah.
So anyway, sir,
So
we will that YouTube link as well. But the only problem is that we were not able to hear any voice from the ma'am's.
No. I'm quite quietly. I'm quite happy with what you give me.
So thank you, sir. Thank you, sir. And means the thing is that we have another session at 4 o'clock again. So I think
I saw that. That's why I was going to tell you. 4, I'll reach airport. So except for the security business, and I'll be able to continue because my flight is only at 5:30.
Yes, sir. Yes, sir. Yeah, so ma'am has joined. So I think we will continue because at 4 o'clock we will be having another speaker. So we will.
That is also a very good topic.
Yes, sir. Yes, sir. Yes, sir.
I understand. Am I audible now?
Yes, ma'am. As of now, you're audible, ma'am.
Okay, and the screen is visible?
Yes, ma'am. Yes, ma'am. It is visible.
Okay. So coming to the helpful if a coach games. These games is another way of involvement of digital technologies for emotion regulation. Young people, user engagement, motivation, helping. Similar technique, gamification is used when different elements of a game is certain sections in a series through several levels, so it helps children manage their frustration, learn emotion regulation. Virtual reality is another area where interactive stimulating a specific towards augmented reality like Pokemon Go and all. In mental health research, headset-based VR has been used for exposure therapy, mostly studied with adults. In one study, which is known as ProReal, teenage found that it helps them to express themselves, help them to externally communicate and their experiences. And recently, I attended one conference where someone had demonstrated and had exposed it one VR for very, very interesting work where they were using working with fear of death by using a virtual reality and making videos, which are very popular way to provide mental health intervention in form of self-help tools for tracking and monitoring one's symptoms, one's mood, one's understanding about one's emotions, or education about it, and to learn relaxation techniques. But the limitations is that although thousands of such apps are available, majority are not tested, evaluated, or may not even be developed on sound theoretical underpinnings. Those which have been studied may not go beyond a publication or report. So, and then some reviews also indicate that there is less use, limited evidence of its effectiveness, especially in the long term. Although when those who are receptive towards it, they also may have some kind of hesitation due to anonymity, privacy, availability, concerns about trustworthiness, privacy, etc. Online peer support is another way where people come together.
Terminal 3
इस तरह 3 यही होगा क्योंकि वो भी टाटा फैमिली है.
Sir, can you please mute?
T3 से गया है. तो आप ये पूछ रहे
another way where they find that our first contact help is usually our peers or similar people who are in our age group and all. So internet has provided a way for people to come together, like message board, groups, forums, etc. And people gain mental health information, advice, emotional support. However, there are concerns for these naturally occurring forums because of its effect on what kind of advice is provided and what kind of effect it might have on mental health and well-being. However, there are also some studies where they look into trained mental health professionals who might be providing these supports, or moderate some of these forums and all, where people can get emotional support, feel less isolated, and there is an opportunity for knowledge exchange, and also the responses are being moderated. Although there are some limitations also where there can be harmful ideas given, or people can be experiencing bullying or negative comments. Online counseling, it can be synchronous or asynchronous. It's also very popular now, especially at post-COVID. Apart from that, there are trained listeners also available. But there are concerns that therapeutic alliance might be are different in these online counseling forums or ways. And here comes some of the, as we had discussed, the AI-based cues which can tell the therapist about how the person might be feeling and modify or adapt their therapy in an online counseling based on these inputs. So computer-based cognitive trainings are other options which are for working memory, cognitive biases and all. I'll go a little faster here. There are certain very interesting concerns. For example, there was one chatbot which was used for people having eating disorders. But then this chatbot started offering dieting advice to the person. And then this was a university research project, which was then ended. They stopped the chatbot because it led to some fears that, okay, a person having an eating disorder, when they are told to diet further, it may actually backfire. Another area where there's a lot of work suddenly is happening is related to death, especially post-COVID. So there is some company which is coming up, which is trying to work with a grief and AI-based grief bots, where in fact, one of the model is where they are trying to use the loved ones' old chats to make an AI bot which mimics the responses of a person. But that is also bringing various kinds of ethical concern about, for example, a person's consent, because it was not taking the consent of the person who is dead now. Also, whether this might actually lead to, you can say, unhealthy attachment or clinginess. Then there are other concerns related to biases in the data on which AI was trained. For example, in one study where they asked AI to create images of a Black African doctor treating white kids, which did not happen actually. In fact, most of the time it showed the doctor as the white and the children as the Black. Another concern which is coming up also is related to preserving humanity in the age of robots. And then finally, which I wonder many of you might have watched this movie, Her. People falling in love with or wanting to have relationship with a chatbot, which might seem like real to them, however, may lead to a false sense of empathy and intimacy, which is not present there as compared to a real relationship. And it can actually lead to more loneliness of people and social isolation. One WHO study in 2023, they looked into AI's use in mental health, and they found that there is the unbalanced research is present because most studies have been done in depressive disorders, schizophrenia, and other psychotic disorders. There are significant flaws in data management because how it processes statistics, infrequent data validation is present, little evaluation of risk of bias, inaccurate interpretation of results. Lack of transparent reporting on the AI models are also there because data and models mostly remain private, and replicability is a question. And then possibility of over-optimism about AI and its accelerated promotion without pausing to assess real-world viability. With this, I will end. The interaction of human emotion technology will continue. Both has positive and negative effects are present. There are many ways to use it for human welfare, which will continue with newer technology, but there's need for more work, and especially in the Indian context. Thank you.
Thank you very much, ma'am. And it indeed a very insightful and very important session for all of us. And we are leaving this stage for the audience in case of any questions. May please unmute yourself and ask for the questions, please. Any question? Okay, ma'am. I had always in my mind the very basic question about in case you can put some light, how those affirmations, positive affirmations, works in the well-being and the behavior of the person. Is there any scientific evidence of those? Does that work actually?
Yeah, so positive affirmations do work, but it depends. So many of the things cannot be used like a one way to handle everything, because it can also backfire for someone. For example, they feel that I'm using all these positive affirmations, but nothing is happening. So what is their cognitions behind using these affirmations? What is their expectation of using these affirmations? How are they using it? Whether they are using it to deny the actuality of their emotions, not working with those emotions, for example, unpleasant emotions, all of that can come into picture. So positive affirmations does have a role in along with many other aspects of
I know, because that the personality itself is a combination of lot many things. It is not only about DNA, it is not only about the environment, it is not only about the neurological aspects. So I know there are many parameters which is building a personality. In the some way, the affirmation also would be playing the role accordingly, I suppose. In case there is no another question, then we should thank you, ma'am. Ma'am, thank you very much for this very important session. And we are looking forward because there are things which we are working as a technical front, or we are trying to achieve some of the objectives. Those are at their end. But we are not going to go ahead, and we cannot go ahead without your expertise and knowledge, because there are so many things and unhidden, unknown challenges are there in the theories and in the practicals which we need to understand. So ma'am, definitely will connect you again for more collaboration and more insights and more knowledge from you, ma'am. Thank you very much for your time and efforts, ma'am.
Thank you, Dr. Priyanka. It was a very interesting connection with you and rest everyone. I'm sorry because of the internet issues in between, but I hope the slides helped you to understand. I'm especially interested in anyone wanting to scale up some of the work related to what is called Indian psychology, or the ancient Indian psychological insight for mental health, and taking it further. And as I was saying that this AR based work which I was telling, so that was actually being done in Brazil using the Tibetan book of death and dying. So using some of the meditations from there, they were developing this AR and VR this thing technology for helping people to overcome their fear of death. And that is something I found very fascinating, because we have so many ways of working with various kinds of issues from our Indian tradition, that I would be happy to take it further and maybe discuss with anyone who is interested. Thank you, everyone. Thank you.
Thank you, ma'am. We'll get back to you, ma'am, on this topic, and we'll surely work with you, ma'am. Thank you. Thank you, ma'am. So, hello, everyone. So we are here with the second session of the day. This is the last session, and I'm happy to inform that we have Dr. Arpan Banerjee among us. And we welcome you, sir. And a little word about you, that Dr. Arpan Banerjee is scientist F, that is professor in NBRC, National Brain Research Center, India. He is currently holding a profile with as a principal investigator of Cognitive Brain Dynamic Lab housed in NBRC, Manesar, India. His trained research team of more than 40 people adept in the performing cutting-edge research in brain dynamics using EEG, MRI, MEG technologies in last 10 years, which has resulted in several publications in top-tier international journals and conference presentations. He has received prestigious grants from national, that is from DBT, and international bodies like Bill and Melinda Gates Foundation, and achieved deliverables committed to the respective government agencies. He mentored several PhD and MSc students, postdoctoral fellows, and research assistants, some of whom has taken up prominent position in industry and academia and have obtained prestigious fellowships like NPDF and CSRI, that is from DST Government of India. These are the only few words about the sir, but in my personal experience, we found he being the true mentor of us, because when we started our work in this domain, so Dr. Arpan Banerjee was the one name where we can, means anytime we can go and ask for any help, and every time he came up with the guidance, and he provided insights and knowledge, and he supported us on all the aspects, all possible aspects. So he is a true mentor and guide for us, and we are looking to hear from you, sir. And thank you for joining us for today. And stage is yours, sir. Thank you, sir.
Thank you very much, Dr. Priyanka, and that was very kind and introduction. I don't think I have done enough to deserve that kind of introduction, but we'll try to sort of forge a more stronger collaboration over the years. And thank you also for bringing me to this meeting. And I think it's a important and timely discussion meeting that is going on. We need a lot of computer scientists and engineers to help us neuroscientists in understanding the brain in a much more quantified way. I just hope that my voice is clear to you and rest of the group.
Yeah, pretty clear, sir. It is loud and clear, and it is
okay, very fantastic. So I will try to see if this presentation, I'll try to share my screen. Let's see how this works.
Sir, there is a 4 button from left in the bottom.
Yeah.
Yeah, we got
Can
it,
you
sir.
see my screen?
Yeah, we got it, sir.
Okay. So as you sort of see, the title is that for today, because I'm talking in front of a lot of engineers and computer scientists. So we will stick to more of the computational aspects that I have been working on over the years. So the title of the talk is AI-powered virtual brain models for health and disease. For this crowd, I don't need to introduce what I mean by AI. I think people are well familiar. Most of you know these terms and all the techniques better than me. So, but I would just like to point out to the complexity of the problem that we have to deal with whenever we talk about anything, any use cases in neuroscience. which is this complexity of multiple spatial and temporal scales. So this is, I kind of borrowed this picture from a nice review article in Philosophical Transaction of Royal Society by Richard Frakoyak and Henry Markram, who you might have heard his name based on the Blue Brain project. He's the sort of the brainchild of the Blue Brain project. And sort of, the idea is this: on the left, you see a lot of structural scales, and going from the proteins and all the way to the scale of our effectors, which is the hands and legs. And on the right, we have the functional scales, which goes from the molecular dynamics of the proteins to all the way to developmental factors such as aging, which, by the way, is one of my core interests, particularly how the human brain and the body develops over the time to sometimes, retain some incredible functions, as well as sometimes retain some functions which get better with age, which is very rarely seen in other species in the planet. So to cut the long story short, this review talks about the challenge for neuroscientists, and I mean everybody, by that I mean all of you, is to bridge these scales. How do we understand, first of all, patterns at each of these scales, and how do we make sense of how these scales are organized and interact with each other? Because if you are looking at one scale, you are not going to get a complete picture of the brain dynamical complexity that is underlying it. So you really have to look at multiple scales, and not only that, also the interaction between the different scales. So if you go into neuroscience literature, just to give you a brief sort of outline here, that typically there are different domains of neuroscience, such as cognitive neuroscience and systems neuroscience and molecular neuroscience, which you might have heard of and maybe wondered that what do these things really mean. So when we talk about these different kind of neuroscience subdivisions, it's actually referring to these different scales, which are sometimes orders of magnitude different. So if you go from molecular to the system scales and then to the cognitive scale, which actually appears when we talk more about functions. So even if I draw these scales, the boundaries of scale, these scales are not really very well defined. There is a lot of overlap which you can find between the systems and molecular, and the same with the cognitive and the systems at both the spatial and the temporal domains. So how do we record such complexity? Well, one of the ways that a lot of scientists do is kind of record from single neurons, so which is at the system, like very low bottom of the system scale and maybe close to the molecular scale. And then we can also, and so which means you can record these action potentials and the volley of action potentials, as well as how the two neurons are kind of functionally connected, meaning whether the action potentials between a pair of neurons at different brain areas co-vary with each other, which you see in the bottom right left-hand corner via this correlogram. Which essentially means that this firing can be correlated across different neurons. This is what this picture displays. And as you go along, if you see that there is, if you look at the sort of, lower frequencies in the extracellular recordings, the electrophysiological recordings, we get this kind of smoother sort of activity, LFP, we call them the field activity, which are actually summation of the extracellular activity. So they are not really one action potential, but they are really summed up input to the cell, which we are measuring via the LFP, and then at a very large scale at EEG, which also looks similar. And we can do a lot of this signal processing. We can apply a lot of signal processing techniques such as Fourier transforms, such as wavelet transforms, to look at how power fluctuates, and this is what is seen at the bottom. So you might see some figures like this appearing in my talk, which essentially means I'm looking at the power of the signals at different frequencies as they vary with time here. And these hotspots typically mean the power of the frequency at that particular location of frequency and time is high, and when it goes to low, then again this kind of goes lower. And the next, another scale that we can measure, and that is also we can measure through fMRI recordings, which is this functional magnetic resonance imaging data, where we can get a very high spatial resolution. But the temporal resolution, which is appearing at the millisecond level, or so to speak, the speed of thought, that we typically miss in the fMRI signal, which is actually, again, as if from an engineering perspective, if you can think of, this is like a huge filter, very low pass filter you have put on the data, and that's what we typically get, which are shown in the bottom here, and that appears in the order of seconds. So the question is, we already have even at the systems and in the cognitive scale, multiple different scales, from the way of single neurons all the way to the fMRI. So how do we sort of bridge this kind of technologies to make sense of the data? Now, what do we really want to make sense about? So what we want to make sense about is the behavior of an organism, in this case a human, such as particular measures such as reaction time. We can measure accuracy of a task. We can also measure subjective reports of perception, whether you are seeing a particular illusion, for example, or not. We can measure, for example, if you go to the neuropsychiatric disorders, we can look at different kind of cognitive batteries. We can look at anxiety depression scores. We can look at, for example, learning disabilities, autism. We can look at IQ. We can look at cognitive and emotional scores. We can do, for dementia, we can do mini mental state exams and all that kind of stuff. Now, the key is that how do these measurements of behavior are related to the neural variables. That's what we are interested in. And we can record all these measurements, as you can see, at the level of both structure, meaning that we can record at what are the detailed pathways that connects between different brain areas. For example, whether a person who is a very intelligent person has a distribution, statistically distributed, stronger connections, meaning thicker set of fibers between different brain areas or not, and so on, so to speak. And then we come back, we can look at also the function, meaning whether the brain activity of that person is strongly covering with other kind of brain activity of another region. So this will be some sort of a functional measure. So we can look at both the structural measure, so we can look at both the functional measure, and we can look them at different spatial scales and different temporal scales. So essentially, this is all these dimensions give rise to the complexity of the data. So for to simplify it though, however, for the rest of the talk, I will use this kind of techniques to, or representations to determine, represent the structural and the functional complexity. One is we can take the data from diffusion MRI, which is a technique in MRI, through which we can record from all the fibers that connects different brain regions. We can compute a matrix, which is the fiber density matrix, which essentially means how thick the fibers are within an individual brains between two candidate brain regions. So if, let's say, one brain region is represented in an entry in this matrix, the connectivity of it with any other brain region will be represented via a the color bar here in this image SC plot, where we can look at, so for example, the red one means that the fiber set between connecting this area and this area are very thick, versus the lower blue and towards the blue means they are very thin to somewhere where it is dark blue, there is no connection. Right, so this is what I would call then is a structural connectivity matrix. Similarly, we can, like we have done for the density, we can do it for the distance, which means whether the fiber is long or short between two different regions. So together, we can get a summary of these in a structural connectivity matrix. Similarly, plot like this can also be made if I represent the Pearson correlation pairwise between all different possible brain areas that I have imaged. And typically in an fMRI experiment, we can do that across different voxels. Typically, if you have a 1 millimeter voxels, that will be 5,000 voxels. So it should be a 5,000 by 5,000 matrix. So what researchers, as you know, as computer scientists, 5,000 by 5,000 matrices are very difficult to invert. So we want to reduce that to a much more tractable size. So we can identify different brain locations with what we call as a parcellated brain, meaning we can reduce the complexity of individual voxels to majorly known brain areas, let's say visual area, auditory area, speech area, and all that. And that way we can make these matrices much lower in size, maybe in the order of, let's say, 60. Some people have gone up to 1,000 is necessary for looking at very specific functions of certain brain areas. So dependent on how specific you want to go, you can increase the size, versus how general you want to be, you can reduce the size of this matrix based on certain transformations. And good for us that there is a lot of atlases available. So these are basically some sort of matrix multiplication at some level. So not very difficult to do with a powerful computer. So, however, now these matrices that I talked about, these are structural and functional connectivity, are what we can get from the brain data. But remember, the parameters from the perspective, if we really want to transform it to a clinician or a biologist, we want to really look at something much more biological. So what is biological? For example, if let's say some disease like multiple sclerosis, which is an inflammation of the myelin sheath, and I will talk about it more in the talk, we can be interested in nerve conduction speeds, because the nerve conduction speeds deteriorates in this disorder as we go along, as the myelin get more inflamed and not. So which means that the neurons are not communicating fast enough. So nerve conduction speed can be one sort of parameter of interest, and you want to see, for example, a drug target can increase or decrease the speed. You can do synaptic scaling, for example. You can measure this you are interested in measuring because you want to look at that if you want to study memory or development. You can look at, if you are interested in psychiatric disorders, different kinds of neurotransmitters such as dopamine, acetylcholine, GABA, and glutamate. Now, the question is, we can very well do this in animal model. That's why you see a lot of research has been done on animals, that we then inject something to these animals and look at their brain activity and see how this, changes their behavior. But of course, you cannot do this in humans. What you have in humans is fMRI, EEG, this kind of recording. So one of the main question or the research that we do in the lab is to see if some of these parameters, which are of clinical interest, which are deep down at a different scale, can be extracted with the power of AI from data which is appearing at a very different scale, which is spatial and very far off from the source even, right? In the case of EEG or MEG in particular. So the way we would do that, we would we would try to sort of develop a computational model, and this is something, an overbearing sort of approach, that overarching approach that will be followed in the rest of my talk. So we will have a model, which means that we can actually come up with a mathematical model of how the brain literally works, or at least local region of the brain works, and then we can connect them using this kind of modes. And this modules itself may be a computationally complex sort of entity, maybe a set of differential equations, maybe a set of partial differential equations, maybe a set of integral equations, but they can be connected to another set of module which will have a similar kind of complexity or something more, even more, across the entire brain. And then we run this kind of code, this kind of programs in a large supercomputer to deduce about the complexity. We look at the functional complexity, and we look at, then what we do, we look at the empirical data. And from the empirical data also we can compute these quantities. So one of the quantities that we can compute, for example, is phase locking, which means we can look at the relative phase of these across different brain regions. So phase, everybody knows, if you have a oscillatory signal, you can compute the amplitude and the phase. Turns out the phase is a good measure of how synchronized. Relative phase can be a good measure of how synchronized they are. So we can look at this phase blocking across the entire brain, and then we can constrain, meaning from the simulated data, I sort of, simulate the phase of the brain area, and which is in a piece of computer in silico. And then we also have the empirical data from fMRI or MEG or EEG, and we can sort of map. So basically do a least square feet, and that's where AI comes in. So AI can help us guide these parameters. So you can extract the parameters which can be then put as a free parameter in the model, such as the GABA concentration, the glutamate concentration, such as synaptic scaling and all that. And we can deduce this parameter based on a particular sort of, interest sort of theme or a certain use case that we can then use in that particular disorder or disease, making an understanding, making or treating a drug, or developing a drug target, knowledge for a drug target and all that. So, but you have got to remember that, you can tell me, okay, this is fine, this sounds very interesting, but the complexity is humongous, and the parameters, the estimation that you have to run out, is actually quite incredible, and it is probably not feasible to do. And you will be absolutely right in saying that. So one has to be very careful, very specific, and very sort of, have that insight at where to apply these kind of models. So it will be applicable in a very narrow domain, in a very cohort of populations where you have relevant data from. For example, age matching is crucial. Maybe looking at their sort of, other parameters of the persons, the weight and all that can become an important factor when you kind of test or train your AI system, whichever training data you feed in. Your prediction will be restricted to the limitations of that data. So that will always be there when we do this kind of modeling. So nonetheless, so this is my observational scale, where we can get these observations of phase locking, the measures, and one of the measures is also metastability, and I will not go in too much on this, but this is another measure that we have been developing for some time now in the lab, which actually works very well for slow time series such as the one that you get in fMRI, because it kind of gives you a measure of the dynamic repertoire of the brain, which means that the complexity of the temporal complexity of the brain can be captured by this measure. So I will just make the next slide what I would do. So this is basically the earlier slide. So essentially from a computer scientist perspective, a bird's eye view of our approach would be the following. So we have a set of big data. We can actually include a lot of transcriptomics, proteomics going in the future, and we can build a sort of network, which are these neural networks, and you can add the neural data also in this column here. And so from there, we can build these networks, which are the networks, understanding about how the network is processing a particular information. And then we develop these algorithms, which can be used for prediction of certain kind of certain kind of patterns, which in particular will fit the data the best to an observable measure. So this is we can do for training, and then using the kind of AI tools, we can go for the prediction of these parameters. So this in a nutshell is the approach that we will follow. So how do this work in particularly, let's say, a use case of DTI or to fMRI? So as I said, so we can compute this MRI data. We take the MRI data, which is essentially the DTI data. We do so the AAL is one kind of parcellation. This is roughly around composed of around 118 brain areas. So instead of 5,000 by 5,000, our matrix becomes smaller, like 118 by 118, which can be easily portable, and we can use simple, like our laptops to sort of do a lot of nice analysis and generate figures. So we generate a structural connectivity matrix such as these, where, as I explained to you earlier, entries like means the number of it can be thickness, it can be distance, depends on whatever you are interested in. Then we go to the functional measure. Now in the functional measure, we can look at the voxel time series, compute the correlations, and these heat maps represent how these two areas are kind of functionally connected. Again, we kind of transfer that via AAL through this FC matrix, which one is we call structural connectivity or SC matrix. This is what we will call it as FC matrix. RS stands for the resting state, which means the connection of functional data when the brain is at rest and not doing any task. And turns out that is an emerging piece of literature, that a lot of work can be done with the resting state. A lot of important functions of the human brain are actually undertaken when we are at rest, not really overtly engaged in a particular task, such as listening to this talk, for example. So we can do the same thing with the MEG data. With the MEG data, what we need to do is additional step of taking the MEG data and then convert to the source level. And this is a completely different lecture. It has its own kind of complexities, but I will not go there. So seemingly like you think of these, that the data at the very sensor level, meaning at the outside the head, can be translated to something inside the head via a set of algorithms again. And then we can again do this on the AL Atlas, and we again have 1M1 by 1M1 kind of time series which represent my functional data. Now, what is the model that I'm talking about? So, well, we cannot, if you are aware of computational modeling, this all started in the year 1952. A lot of this work actually came to shape first with this Nobel Prize winning work from Hodgkin and Huxley, where they were recording from giant squid neurons, and they kind of came up with this model of membrane voltage, which is where voltage is V, represented V, and these M, H, and N are actually ionic currents or probabilities of ionic currents. These are differential equations which represent, and these you can see they are heavily nonlinear differential equations. And if I solve this differential equation, we can get something like this V as a function of t will look like this, which means an action potential can be represented by a set of this kind of ordinary differential equations which are coupled, which are nonlinear. Now can we put this neuron on one single neuron equation at each of these individual brain areas that are represented with the structural connectivity, couple them, run the code, and can we get the functional connectivity analysis? Well, the answer is no, because these are not individual neurons that are connected when we are talking about the structural connectivity that we are measuring from the data. So these are actually thousands of neurons. So thousands of these guys. So if you imagine that this equation is 4-dimensional for one neuron, for 1,000 itself, like for one local brain area, let's say it has some 10,000 neurons. So there will be 10,000 times 4 number of differential equations. So we can't use that. So to save our lives, so this was the work by, again, Wilson and Kawan, two amazing mathematicians who developed this sort of understanding that the set of excitatory firing. So now if you have a strong enough stimulus to the brain, a bunch of brain areas together get synchronized. And there will be an excitatory set of neurons, there will be inhibitory set of neurons, and the combined joint firing rate of all these neurons can be represented by a common differential equation. So instead of looking at one specific neuron, you can look at the field activity that is generated by several thousand of neurons. And this can be represented by E for the excitatory neuron. For I, the inhibitory neuron current is being represented, or sorry, the voltage is being represented. And now what we can have is a set of combination or a coupled population of excitatory and inhibitory neurons, which will look like this. We can have two differential equations instead of this 4-dimensional 4 times 1,000 times 80,000 differential equation. We can reduce them to 2-dimensional equation as long as the neurons that we are talking about are together firing at the same time. Okay, so this is what we call is a population model of neural activity. And with the population, we are talking about if I dig deep in the cortex, it's just one patch of the cortex that I can model, which consists of a set of excitatory and inhibitory neurons. This is not even far away from the whole brain. Now, what we can do, so hence, since it is far away from the whole brain, we cannot really technically use that either, because this is also too simplistic from the model that I am talking about. So what we do then is that we can actually model, and this is something that has been first done by Gustavo Deco and then followed up with a lot of work from our lab, where we can actually model the currents of these excitatory and inhibitory neurons and we can couple them via the CIJ matrix, which will be the matrix that is coming from actual structural connectivity of the data. And what we can add two sets of equation here, which stands for the synaptic scaling, for example, the variable that I was talking about, how that changes with time. And it also can have the GABA and the glutamate and the GABA concentration as part of parameters of this model. So essentially, this is a 6-dimensional set of equations, and this can be connected over the entire brain. Let's say we have 118 of them for NAL parcellation. So we will have 118 times 6 dimensional coupled differential equation system that is going to give me a virtual model of the brain. And then what we can do, we can tweak individual areas, we can tweak the concentration of GABA glutamate in a particular brain area, or we can even make lesions to compute stroke, like, effects of stroke, for example, stroke recovery trajectories. All these can be done and predicted once we have a good set of training data over this model. So I will give you three. I hope I am convinced you that this is possible. First of all, getting a virtual brain model for the entire brain, and we can set or put our or train our or the imaging data that we collect from EEG and fMRI on this set of models and generate some sort of predictions. So the next what I will do is I will give you some examples of use cases, such as one example will be in health, as I said in the title, that we want to apply it into understand our health. So in this case, the interesting part for us is healthy aging. The second that I will talk about is how is brain lateralized. For example, you might have heard speech is left lateralized or music is right lateralized. How do these lateralization appear in the brain? Can we predict the lateralization of an individual's brain with this kind of models? This I will again show. And finally, I will show you an example of a particular disorder which recently got published in iScience to look at the clinical relevance of this kind of models. So to start off, so this is something that we kind of did for actually quite a lot of subjects. So what we did is that we set up the equations that I showed you in the previous slide, where we can have the 6-dimensional set of equations and model a brain functional dynamics or the fMRI activity. We can take now, we can compute, we can invert this model based on training data set. We can invert this model for a large cohort of people, individual by individual. We can compute the GABA and the glutamate concentration as a free parameter, which we feed by an AI algorithm. And what we do then is that we compute the statistics of these GABA and glutamate concentrations that we predict from an individual brain across the cohort. And amazingly, what we found out is that this is for CAM-CAN cohort, which is a data that has been collected in Cambridge Aging Neuroscience Group. And we have been using their data for a long time. And this consists of 650 healthy, which is a lot of data, in imaging literature, 650 healthy human subjects. And what we can do is that we can predict the GABA and glutamate concentration as a change from young to old. And what you see, what is interesting, we found out when we did the statistics across all subjects, that the GABA is, we got a null result for the GABA, meaning the healthy aging, GABA is not changing. And the age range here goes from 18 to 88 years old of individuals, healthy humans who has no history of cardiovascular disease, for example. Now, the glutamate, on the other hand, the concentration actually significantly reduces. Now, and same thing for the GABA glutamate concentration. So we can look at this because this is an interesting term for biologists, because this determines the excitatory inhibitory ratio, which means the balance between our excitatory and inhibitory neurotransmitter system or excitatory and inhibitory activity in the brain, which governs a lot of homeostatic functions. So what because since the glutamate was changing, the ratio of the glutamate to GABA was also significantly changing. Now we went to another data set, and this was around 300 subjects. This is NKI Rockland data, which is collected in US. This was in UK. And again, lo and behold, the same pattern we found out. We also tested in a local data set, which we obtained from one of our collaborators' lab in Berlin. And again, the same pattern kind of remains salient. So in all these data, we essentially see the same pattern, like over a set of even is more than around 1,000 subjects, we are seeing glutamate is changing significantly and GABA is not. And this result was kind of, we didn't know about this pattern previously. And then we searched the literature once we started getting it in the CAN. And lo and behold, what we found that people using measures like MRS, magnetic resonance spectroscopy, which is essentially a version of NMR, which you can do on the brain to find out metabolite concentration, they reported that the healthy aging doesn't involve a change in GABA versus pathological aging actually involves a change in GABA, such as in the case of Alzheimer's disease, in the case of Parkinson's disease, you do see a huge change in the shift in GABA, but not in healthy aging. And this is something that we kind of tested, or our model also sort of predicted essentially, which these results essentially mean. So we also did it for another kind of data set, though not exactly from a neurotransmitter perspective. We can actually look at these other parameters, like, as I said, synaptic scaling and kind of conduction speeds. And this is actually with the MEG data. This is what this same sort of, cohort CAN also had MEG data along with the fMRI data. So what we did there is look at the phase locking behavior across the brain. And what we found is an amazing result is that at the alpha frequencies, which you might have all heard about, the most dominant EEG frequency at 10 Hertz, as you see, as we grow older from 18 to 88 years old, there is a change in the frequency of the alpha rhythm. From like high frequency, it goes to a lower frequency. And then what you also see, and this is kind of shown very well here, so as you see, the 10 Hertz alpha is decreasing in frequency. What is interesting though, the phase locking behavior, which you can compute by something which we call the phase locking index, is actually unchanged over aging. So if you look at, which means the crosstalk between the brain areas, how they are communicating between them across the entire brain taken as an average, remains completely invariant as we go with age, even though the frequency is changing. So taking an anecdote from maybe a gymnasium where we go on a sort of, on a treadmill, when we get old, we are not able to stay on the treadmill if you set the speed of the treadmill too fast. But you can stay on the treadmill if you decrease it to a lower frequency. The most important point is to be staying on the treadmill, and that's what seemingly brain is trying to do. So it is lowering down its frequency to be balanced, to be communicating with other regions of the brain and preserve a lot of the functions. In the psychology literature, this kind of phenomena is called neurocompensation. And this is exactly what we are seeing through this kind of modeling techniques. So, well, till I showed you the empirical data, we did a model. Again, this is based on a kind of a simple harmonic oscillator kind of thought process, that if you think of each brain area is behaving like these pendulums here, which has a phase, and these phases are now coupled by a structural connectivity matrix, and which are also these differential equations. So these phases are varying with a lag, which across different areas. So these are actually what we call is ODEs, but they are time delayed ODEs, which is the time delay is coming from this delayed part is coming from the actual delays that take for information to go from one point of the brain and to the other point of the brain. These equations are quite complex, though they look simple, because of this time delay term makes this an infinite dimensional system. So nonetheless, we could show that the parameter space can be shown to be that we can get a sort of, this conduction speed and the coupling. There seems to be these isofrequency lines where this speed, as I said, the speed needs to kind of go down to maintain to be on the frequency or the phase locking index lines, right? So these are the kind of things one can do with this kind of models in different forms. So just to give you another use case example of speech and music, I will just show you the symmetry in the auditory system, where you can hear from sounds through different kind of, the left and the right ear, and they cross at the thalamic level. The thalamus is a piece of structure which is not the cortex, but somewhere outside the cortex. And then what is even more interesting, when we look at the brain data, we typically see for speech-related kind of tasks, we see a lot of left hemispheric activation, meaning the Broca's area is typically, when we talk about the Broca's area for speech, it is in the left hemisphere. Whereas when we see, when we present something like a music with melody, it goes to the right hemisphere. And there are people who have shown that this is actually the case. And one of the most elegant way to show this was but before that, I just wanted to mention one more thing here. It's not only the music or the melody, any kind of tonal sound, such as this auditory steady state stimuli. So if you present 1 kilohertz tone like beep, 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 something like that, you present it across the entire time, and these are oscillating at 40 hertz. You see a sort of a pattern emerging in the brain. And what my PhD student Neeraj showed, that if you present it binaurally, monoaurally, or on the monaural right, it doesn't matter. The dominance is always in the right hemisphere. So for example, this is what we saw in this paper, that if you see for monaural left, monaural right, everywhere, the right hemisphere activity of the brain is much more than the left hemisphere activity, which essentially means that it is a right hemisphere dominant phenomena. So any tonal sound, for that matter, anything which is related to just very simplistic mixture of tones, it goes to the right hemisphere. Another way to think about it, something which is required, like sort of processing of tones, which are somewhat in a way a little slower process. This is not our hypothesis. This was given by Robert Zatore's group in Montreal. So they propose that it is really dependent on the external frequency, whether it is the fast time scale or the slow time scale that you want to pick up. For example, speech, which is very fast, you have to attend to the very fast changes in the contours of the spectra. So that's what makes it left hemispheric. And whereas this one makes it, if you are interested in much more slower processing, such as the melody, the timbre, maybe the aesthetics, whether the music is beautiful or ugly. So this kind of resolution requires much more longer time scale processing, which is why these are going in the right hemisphere. So this is one of the hypotheses that is existent in the literature, why these two bifurcation is there. So what we kind of came up with the idea that, well, we should have, if if you present the same stimuli, in one case, let's say something like a sort of a spoken, like sung speech, for example, there is a black crow, something like this. So you present text in the form of music. Now, if I ask you to attend to the sort of the melody aspect, the same stimuli should elicit right hemispheric activity, whereas if you are attending to just the syntax of it, what I meant by the English sort of sentence or the English grammar that was involved in the sentence, if I want you to attend to that aspect, it will go more towards the left hemisphere. And this is what Zatore in a very nice science study showed. So what we thought about, if we do sort of a computational model and we kind of train the model, can we see the certain similar things through the model? So for example, if we give it a sort of, if we have a whole brain model and we give it a fast stimuli, does it go to the left hemisphere versus the very slow sort of contextual stimuli? Is it processed in the right hemisphere? So this is what Neeraj did, but before that he did an EEG experiment to verify what they observed in fMRI is actually varied. And I don't think, I probably didn't share it with the sound, so it may not play, but I'll just quickly try once to show you what kind of stimuli we are talking about. Can you hear can anyone say if they heard it?
Yes, sir. Yes, sir, we could hear.
Okay, good. So you can hear it. So this is exactly kind of sort of, the sound that we were presenting. So in this case, it was the syntax was this: the servant came to love the children's dog, right? So I will just present to you again. So now,
so the sound is just a beep kind of sound. Am I supposed to listen that beep sound, or something else is also singing along with that?
No, so this is the song. Now, based on the task comes later. So
that is very feeble, sir. Means the other sound is very feeble. Means only beep sound is hearable right now.
But the main thing is here. So I think what you heard is some melody. May not be if you were not very clear about the lyrics that was inside there. So what we were doing here is let's say I cue you to tell me that the first stimuli, like I present bunch of time, 2 times one stimuli. Now, in one case, I want you to attend to the speech and ask you, the 2 set of music had the same speech component, right? So you attend to only the meaning of what the person is speaking through the song. And you say whether they're same or different. That is one kind of task. And we can do it in the melody context. Now we have made pairs. So like you go back to that scene, the same music will also be said, they said it was a cross they saw in church. So all the songs can be combined like interchangeably with the lyrics can be combined with the music kind of, fluidly. So the same lyrics can have both these sort of, music, and the same goes vice versa for the music. Yeah. So we can mix them up, and what we can say, based on the attending to the speech or the melody, we can make the task, we can ask the subjects to do that, and the subjects were pretty good in discriminating that, the speech and the melody. So there is more than 90% of discrimination. So the stimulus was shared by Alboy and Zatori, who are also part of later now is also collaborating with us on this. So what we also looked at now is that we can look at this causal connectivity between the brain areas once we go to the source level and using a measure called Granger causality. And in the interest of time, I will not get into what is this. Just trust me on this, that we can sort of determine how each brain area is interacting each other, actually the arrows, which is beyond what normal correlations can do, which is what is you need to kind of keep in mind. So we are going beyond now using this measure, what normal correlations can do, which is the correlations gives you covariance, meaning a statistical similarity between directions, but you cannot draw these kind of arrows. With this measure, Granger causality, you can actually draw these arrows. We can see which brain area is causing a change in the other brain area. Yeah. So using that, what we can do is that we can actually look at we can set the seat at primary auditory cortices, and we can look at how primary auditory cortices, which are situated at the temporal lobe here and here. So we can look at how this so this entire area is parcel rather, which if you take into AL, this will be represented at one data point. This entire, all these brain areas along this colored space, and again this colored space in a different point, and how these are projecting to the other brain areas. So what we can divide this entire frequency, since this is EEG, we can actually divide it into different frequency band, for example, delta, theta, beta, and gamma. And what we can see that for each frequency band, whether the connectivity from the auditory cortex to the rest of the brain was left dominant or whether it was right dominant. Okay. So this is a bit sort of complex sort of map. I think you have to visualize. It takes some time to get a hang of it. But to cut the long story short, what you kind of, just see that in some sense that, delta and theta, what you are seeing for speech, you get in a lot of these band, well, for the delta and the sort of, band and the theta band, it is more compared to the gamma band and the beta band. But it is more sort of stronger. The activity here, delta band, there was nothing in the right hemisphere for speech. But it is actually much sort of it is very present strongly here. Theta again, it is again left dominant, which is for speech. But beta and gamma, they are not really, they are kind of bi-hemispheric. So both the hemispheres had equal representation of this, directional coupling in beta and gamma. For the case of melody, an opposite sort of effect happened in the delta and the theta bands. So delta and theta is that we are talking about the power here in this spectrum here. Yeah. So, and in this case, in the melody, you are seeing the right hemisphere is more active than the left hemisphere again. And the same pattern of results you get, the beta and the gamma seems to be more bihemispheric. So the hemispheric lateralization of speech and music, first of all, it happens at lower frequencies, and it can be visible with EEG, which is something that is important to look at. And this, we could also replicate our earlier auditory steady state rhythm in this set of cohort to make sure. In the next set, what you see here, as I said already, that if I plot this hemispheric lateralization, imagine I can compute the hemispheric lateralization simply by looking at left hemisphere activity in a brain area minus its right hemispheric analog divided by left hemisphere plus right hemisphere. So that gives me, if I am right dominant, I will get a positive number. If I am left dominant, I will get a negative number. As you see, all these different areas are ordered, and typically all of them are for the 40 hertz. They are kind of, here they are right dominant in the case of the orbitofrontal and the parahippocampal areas, which is again the tonal sounds. For example, for the speech and the melody, on the other hand, the same areas are beautifully in the delta band. The same analogous areas are right dominant for the case of melody, whereas for the speech, they are left dominant. So which is kind of goes away a little bit in the beta and gamma band. So the previous plot now plotted more in terms of quantitation, so quantifiable numbers of lateralization index. Now, again, we can do this whole brain to kind of, functional structure to function kind of mapping. We can put this kind of oscillators and look into that. And this is where AI comes in again in this picture. So what we want to do from the data, we want to fit the data with the most dominant frequency F, the time delay and the K, based on which the time delay term that here, if I make it a free parameter, this time delay measures the time scale of processing. May measure if you are a very short time delay, that means that it is very fast processing. Very long time delay means it is kind of a longer time scale of processing. So once we go to this, sort of, I will avoid complexity and get to the sort of the main result here. So in the interest of time, what we kind of found out for the melody and the speech, again, the time delays seem to be different. So remember what I told you earlier, one hypothesis in the literature is for the melody, you would have to integrate over a more longer time scale. For the speech, you would have to be faster. And when we look at the time scale of the time delays of this model, when we set up the entire brain to be an ordinary, the phases of the brain oscillators to be something coming like from these what we oscillators we call the Kuramoto oscillators. This time delay seems to be a key parameter which can sort of capture this dichotomy of hemispheric lateralization. So if you see, for example, the melody task, the deduced time delay, the functional time delay that we were getting were much higher, about 10 millisecond difference. So which is quite huge in the case of in terms of the neuronal time delays are concerned. So which we kind of consistently find, and this we can deduce individual subject wise. This distribution what you are seeing is computation of these time delays at individual subject level and looking at their statistical distribution. So this is something that we can do in the healthy brain. As again, we can go to this Zatorre and Belling's hypothesis, and I think we have shown how mechanistically this can be also verified by this such kind of models. So just to for the next 5 minutes before I end the talk, I just wanted to show that I have shown you two kind of scenarios where we can apply these on healthy sort of human data, but are these of clinical significance, as I have pointed out that we should try to achieve. So one of the things that this is something, again, a data, this is MEG data, which is magnetoencephalography data from a cohort of people, 50 individuals who have multiple sclerosis, which is a disorder of the myelin, in which the myelin gets inflamed and it starts off with cognitive symptoms. Slowly it increases and eventually the person gets immobilized at some before the person dies. So it's a kind of neurodegenerative disorder, and it happens in this period of 2 to 5 years. So it's a kind of in a way deadly disease. So what we see here is that, you know, data from a control group of people, and these are early stage detection of multiple sclerosis, and these are the controls are of course the healthy humans who have a very high alpha, the amplitude is very high. The patient, on the other hand, have a very low alpha and the amplitude is very low. The peak frequency, remember, which we can measure, which is at typically alpha is around 8 to 12 hertz. This peak frequency for the control, as you see, varies over a longer range. And here for the patient also, it varies over not so significantly different range. What is only changing, so in some sense, unlike the healthy aging, but the frequency of the alpha was decreasing. In the beginning, I showed you. Here, the amplitude of the alpha is decreasing, but not the frequency. So it's not slowing down in terms of the overall alpha frequencies. However, the area under the curve is also obviously sort of, you know, decreasing significantly. But main thing is there is an amplitude reduction. How do we explain, and can we determine the underlying, is there a change in the delay, the nerve conduction speed, that actually this paper that I'm talking about, this figure is from, is now published in iScience. It came up this year. So what we can do is we can actually model this again using a sort of what you call is a Stewart-Landau oscillator, because we are interested in both the amplitude and the phase here. So we had to go to the complex sort of a valued sort of a, so this is actually a two-dimensional equation system for one brain area. ZZ is the complex sort of distance variable, which can be sort of untangled to have both the phase and the amplitude part. So these are, you will get, if you untangle it, you will get a phase equation and an amplitude equation, two coupled differential equation, and then you connect it over the entire brain via this kind of connection matrix defined by the CJ terms. And these are also a normal form of Hoff bifurcation model. So typically they display this kind of behavior, which means that they are from a decaying spiral to it kind of goes to in the subcritical range. It can form a new set of oscillations in the supercritical range. So we have G positive versus G negative, which is shown here. So now what we did is, once we fit this model on the data, what we deduced, so this term, the DJKV, which also stands for the time delay tau, what you can look at this, and that, you know, and this particularly this MS cohort, what you can see, the patient has a much shorter V, which is the conduction speed, which gives rise to a longer time delay, which means the person will get slower, and this can result to some kind of cognitive impairment. And what we kind of then did was we looked at the cognitive scores of this patient and then correlated them with these time delays. And as you can see, typically if you use any other measure such as gender, age, duration, lesion load, this is typically the established clinical measure, which means that using an MR scan, the doctor determines which part of the brain is inflamed, and using that and how much of the area is inflamed is used to determine as a prognostic marker that how the patient is going to deteriorate. But it doesn't always hold true. For example, that this is known as the clinico-radiological paradox, that even if you have a large lesion load, some MS patients show very less decline of cognitive impairment. They are perfectly fine. Whereas somebody with a very small lesion load, like MS affecting a very small area of the brain, they have a very difficult sort of time. They have a very stronger cognitive impairment. However, when we kind of correlated them with all these markers of cognitive impairment with the estimated nerve conduction speed, which could explain the variance in a much better way. And this is something that is incredible, because this change, what we are getting compared to the normal clinical measure, was almost twofold. And this is something that we wanted. This is now patented. So this is something that wants to be, we want to use for identifying and looking at the prognosis of the multiple sclerosis patients. So I'm just giving you some examples of how this can be done and what are the potential of such kind of modeling, this kind of modeling detailed models of the brain using multi-scale approaches can get you, which is a very rich insight about the underlying biology, as well as we can use it to make clinical sort of applications which will be very useful in the future. So as I said earlier, so this I have already given you some of the pipelines. This is something that we can, anybody wants to use some of our pipelines are welcome. We have the codes with all our papers in the Nashkar et al. Paper as well as the subsequent papers, all online in Bitbucket. And if you still have problems in using our code, you can always ping us, and we will help you how to use them. And with that, I wanted to thank a lot of people who have actually done the work that particularly have shown, I have not named two students who have done the work. That one actually was a postdoctoral fellow who did the aging work. The neurocompensation work on aging with MEG was done by Anak, who recently moved on to a postdoctoral position in France. Who was my PhD student, and Neeraj, who is now a postdoctoral fellow in the lab, did the lateralization work, and also the multiple sclerosis work was done by Anand. And also we at the several of my collaborators, Dipanjan, who is in IIT Jodhpur, Anil, Abhijit, Gustavo, Roslyn, Bina, Dr. Bina from CMC Vellore, Pratap Sharan is also a clinician, neuropsychiatrist, who has been helping us in different understanding of neuropsychiatric disorder from AIMS. Also the different sort of resources. NSG is a good platform to run our codes. We have a collaboration with them, and they have a supercomputing facility. They're based in UCSD. CAMCAN provided us all this data. Also the Human Connectome project, the Child Brain project, which also gives this NKI Rockland data. Open Neuro for other data sets. And thank you very much for attending and listening to the talk. I'll be happy to address any questions that you have.
Yeah. Thank you very much, sir. And as usual, it is very important, very scientific, and very insightful session. So the stage is open for audience. May please unmute your mic and can ask the questions, please.
Hello, am I audible?
Yes.
Hello, sir. I'm Akanksha. Actually, I had one doubt regarding in one place you had covered about inhibitors. So, how exactly do we analyze which are inhibitors and which are not?
So I cannot follow you properly. Some of the words are missing. Can you be a bit louder?
Yes, yes. Am I audible now?
Yeah.
Yes, sir. So I was asking that in a slide you had covered regarding inhibitors. So I wanted to know that how do we categorize them as inhibitors? Like how do we see that?
Okay. I see. So these are we cannot see how many of these excitatory or inhibitory neurons in each brain area are there. Okay, so because from this we cannot measure excitation inhibition separately from neuroimaging data. So that's why where the model comes in. Also, a lot of literature from human cadaver studies and also animal studies say that the excitatory inhibitory ratio is around 1:3. So for example, 50 inhibitory neurons corresponding to 50 inhibitory neurons, there will be 150 excitatory neurons. So we use that kind of insight for set up our area. So each area will have 200, let's say, excitatory and inhibitory neurons. So this is how we kind of choose it. We fix it based on this knowledge, rough knowledge from animal literature as well as cadaver studies in humans. Okay, but they are there. If you measure in single neurons from animals, you will see the excitatory and inhibitory neurons very well. So inhibitory neurons will be the ones in which action potential goes down with the stimuli. Excitatory neurons, the action potentials, the rate of action firing rate will increase with giving the stimuli.
Okay, sir. Sir, one follow-up question. Sir, is this excitatory and inhibitory related to decorrelation in some way, or is it a
Deco-
different angle? Yeah, like we are talking about, say, correlated firing of
No,
neurons.
no, no,
So
no, no. So
the
see, excitatory inhibitory is independent of synchrony. Okay. So excitatory and inhibitory will be there so that that brain doesn't become a runaway, like firing rate will forever keep increasing, right? Of neurons. So if you want to bound it, so something is always, so there is always a balance. So this is one of the things that, say for example, that you see a EEG spectrum looks like this, right? So, but there is a lot of baseline data. So one of the, right now the hypothesis is the baseline spectrum of the EEG comes from this EI balance. So balance firing of excitatory and inhibitory neurons. So they produce this, like, some sort of a homeostatic, they keep the homeostasis in the brain going. Otherwise you will have, epilepsy or something like that if it is
okay,
out
okay.
of balance, where you will only have everything is being synchronized. So excitatory inhibitory somehow balances each other at every different level, be it at the neurotransmitter level or maybe at the firing level. And we are still like several people are trying to figure out what is the nature of this balance.
Okay, sir. Okay, thank you so
So I think there are a couple of questions in the chat box. So one of the question is, can it be early detection of disease like Parkinson's? Yes, since we can add a third term in addition to GABA and glutamate, we can have a dopamine term. So we can look at dopaminergic neurons also, and if you can have some Parkinson data to train the concentrations of dopamine in the Parkinson patient. So then we can once we develop the training model, then we can apply it on any kind of testing model for Parkinson's. And for the healthy brain, it will also make sense to have the the dopamine in the set of equations. One, we have a sort of a training model for that. This is how it works, right? You have to have some kind of training data to know what the pattern looks like in a disease cohort. So yes, if we can generate some data of Parkinson's, in fact, that is something that I did tell one of my students to look at, particularly Parkinson in future. So that is certainly doable. Another question.
Sir, one follow-up question from this: how many subjects we have to? How many subject data we required for training the model?
As many as possible. I mean, 150.
What would be the minimum benchmark for this?
Sorry.
What would be the minimum benchmark for this?
Right. So if you give more diversity of the subjects from different places, from different sort of age groups, from different kinds of like areas, they start having degradation. So the diversity will also help in understanding it better. So the more data you give, the better the training model is going to be. I cannot off the top say how many, where you should do the cutoff. That's difficult to say, right? So once we have to start collecting and do cross validation, then only will we say if the data is good enough or not.
Okay, sir. Thank you.
So there is one question which is observational scale. What is metastability? So okay, so metastability is somewhat complementary to synchronization. So synchronization, phase locking measures how each two sort of systems are sort of synced together. Metastability measures something how far apart they are. Okay, and why is it an important measure? Because that between how much together you can be, how farther away you can be, both gives us a complementary picture of the system. The second aspect of metastability is when we compute the metastability. So for example, from these phase lagged indices, we can compute the vector sum of the phase lagged indices over an entire population of nodes. The metastability will be measured as a approximate metastability will be the standard deviation of the. So if you have a measure whose mean is the order parameter is the mean of the phase lagged index, the standard deviation of that will be of the order parameter is measured as the metastability. So it's kind of a complementary. You can think of mean which gives you something. If you look at the time series, something which is mean versus the variance, which gives you two complementary pictures about the statistics. So metastability is very important measure to constrain the system in that sense. I think
Hello,
there is
sir.
no other question in the chat box. So if you have any other question, please do.
Yeah, sir, I'm Kiran over here. Sir, I have one question. Sir, I have taken one EEG data, time series data for analysis, and I'm trying to build one computational model based on the artificial neural network. Data set consists of depression and anxiety people. So what is happening? There will be the number of patients who are suffering from depression. They also have the background disorder as anxiety. So if I try to build a computational model, my accuracy is going down if I taking both the disorders together. How to sort out such kind of difficulty?
So this is a very good sort of, practical problem. A lot of these computational sort of or the neuropsychiatric disorder has this issue that the symptoms are overlapping. Yeah.
Yes, sir.
So what do we do? So there is so this is itself is the question is, do we have the right measure for the disorder? So then there we have to ask the question, is DSM-5 or ICD-11 or whatever you are using for anxiety, depression, Beck score or whatever, is it the right measure itself? So one sort of way to look at this is we need to develop more better task-based measures. So one of the things that we are, for example, developing there is that we are developing sort of, using music, for example, looking at responses of emotion on the music and looking at the statistics of that. So we have to innovate on on these measure front. So so this is a one serious issue with just blindly using DSM or something like that, and then you will expect a correlation with the brain marker. It's very difficult. You most likely will fail, and that's the the truth of it. So what you would see, a lot of people are trying to develop when they develop their cohort or develop a particular study, a lot of approaches go in determining what metric you will choose or what metric you will use. So your, whatever your anxiety, depression score will be a first level measure, but you need to have a much more advanced measure, which will be much more quantitatively, maybe it will be in the research domain for some time. It cannot be used in the clinic, but that needs to be developed at least to understand these differences or giving it an identity. I do agree, I do sympathize with you, and there is no easy solution to this problem.
Yes, sir. Yes, sir. Thank you, sir. Actually, I'm really struggling for that, how to overcome, because there will be the background of one disorder with the another, and they may go simultaneously also in some patient. So patient to patient also it may vary, I feel. So thank you, sir. It was a very great session, and I really learned so many things from this session. Thanks a lot, sir.
Thank you.
Do we have any other question from the audience? So if it is not, then we are going to close the session and the lecture series as well. So sir, with your session, the series means it is kind of a complete series now, because the aspect which you have covered was really, there were very hidden insights into your session, and it was really we mean to understand. So definitely there are a lot many things which we still are remaining to understand and take your guidance on the points. I may connect you in an offline mode as well. Even the rest of the audience may connect you in an offline mode also. Thank you very much for your time and your session, sir. Thank you for sharing your knowledge. Have a good day, sir. Thank you, sir.
Thank you, Priyanka. Thank you, everyone.
And I thank you all the audience for attending this session and getting the knowledge from the seniors. So thank you all. And this is how we are going to complete our lecture series today. Thank you all of you.